Hi class, we are going to go into chapter 6, which is all about the muscular system. Last week we were talking about the skeletal system. This week we will finally understand how those bones move by putting it together with the muscular system. Let me go to screen sharing mode here, and there we go. So this chapter is all about how our muscles work, and it's a pretty thick chapter when it comes to content. So hopefully you'll have lots of time to listen to me over and over again talk about muscle tissue. So we reviewed muscle tissue a little bit in chapter 3. It's one of the 4 primary tissue types, right? Epithelial, connective, muscular, nervous. It's specialized for contraction, and there's 3 types of muscle tissue. You have skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is noted because it is striated or has stripes, as you see in this picture. Cardiac muscle has intercalated discs. The muscle fibers are actually branched. And smooth muscle has no striations. The cells are spindle-shaped and are not as well organized as in skeletal and cardiac muscle. So again, just a quick review. Skeletal muscle is the main muscle of the body that's providing movement. Cardiac muscle is the heart. Smooth muscle would be found in the walls of hollow organs like the stomach, the small intestine, large intestine, etc. So functions of skeletal muscle. This chapter, for the most part, will be focusing on skeletal muscle. There's a little bit on the other muscles, but for the most part, we're focusing on the general function and structure of skeletal muscle. So skeletal muscle is there for movement, right? There's the contractions, you're pulling on tendons. Tendons are connected to bones, and those bones can then move. It's also to maintain our body posture and our body position. Keeping me upright in this chair are lots of back muscles that are holding me up. They're there to support soft tissue. There's layers of muscle that are holding in your abdominal cavity organs. So those muscles are there holding in your abdomen. They're also there to support your visceral organs and hold them in place, as well as protection. They also guard entrances and exits, like your mouth is surrounded by the orbicularis oris muscle. That's what's moving. I can close it and keep my mouth shut. And there's several openings like that around our body, or we have voluntary control. It's also there to maintain body temperature. When you get cold, you shiver. And those erector pili muscles contract to erect those hairs. So that's all done through the skeletal muscle. It's also there for nutrient reserves. The contractile proteins can be broken down in times of need so that your body can have amino acids for energy. It's not the best source of energy. There's some waste products that are produced. This would be an ultimate starvation mode. People that are, stranded in the middle of a forest or a desert and they're not eating much, they will begin to break down their muscle tissue if they don't have proper protein intake. So let's talk about the structure of skeletal muscle. It's surrounded by several connective tissue layers. The first is called the epimysium. The epimysium surrounds the entire muscle. So if I were to take off your skin and look at your muscle, I would be seeing the epimysium. The epimysium is what's surrounding the muscle. When you're looking at a piece of chicken, raw chicken, sometimes there's a clear layer on top of the chicken. I'm thinking of like the chicken breast or chicken thigh. There's a clear layer that's right on top of the meat. That's the epimysium. It's the outer connective tissue covering. Then you have what's called a perimysium. Now what you have to understand is our muscle fibers or muscle cells are arranged in bundles called fascicles. So the perimysium wraps these fascicles up in little bundles. The fibers themselves are surrounded by an endomysium. So when we start small, I like to start small and get big. When we start small, each muscle cell is called a muscle fiber. That muscle fiber is surrounded by an endomysium. The muscle fibers are grouped together in fascicles. Surrounding the fascicles, the perimysium. All of the fascicles are grouped together to make the entire muscle group and that is surrounded by the epimysium. So those are the connective tissue layers. Tendons are collagen fibers of connective tissue that extend beyond the muscle end. So that epimysium extends or kind of joins together and connects that muscle to the bone. A bundle of fibers is called a tendon. That's, again, attaching the muscle to bone. Whereas if you have a sheet of fibers, you have something called an aponeurosis. This would be a very broad area of connection. You have aponeuroses in your abdomen, on your skull, and on your back. Those are the major areas of aponeuroses on your body. The collagen fibers can actually extend into the bone matrix, rooting them there in place so that they can't move, or they're very hard to detach. So here you see the organization. You have the entire muscle itself. The entire muscle is surrounded by that epimysium. Then you see these little muscle fiber bundles called fascicles. Those fascicles are surrounded by a perimysium. Here you see a fascicle itself. Those fascicles have lots and lots and lots of muscle fibers in them. This one has 1, 2, 3, 4, 5, 6, 7, 8 muscle fibers in this fascicle. That whole thing surrounded by a perimysium, and each individual muscle fiber is surrounded by an endomysium. Here's where it gets trickier, because in each muscle, oops, sorry, I didn't mean to move that. In each muscle fiber, or in each muscle cell, there are myofibrils. Myofibrils. Myofibrils are the contractile units that are within a muscle fiber. And those have their own function and structure, which we'll talk about later. So let's talk about some special terms dealing with the skeletal muscle. First, a muscle cell, as I've said, is called a muscle fiber. The cell membrane of a muscle cell is called a sarcolemma, very different from the typical plasma membrane. It's called a sarcolemma. The cytoplasm is called a sarcoplasm. So that prefix sarco is referring to contractile cells. It's referring to these special muscle cells. Endoplasmic reticulum, or ER, is called sarcoplasmic reticulum, or SR, in muscle fibers. So muscle fibers have several hundred nuclei. Skeletal muscle fibers have several hundred nuclei. They all have these cylindrical structures inside of them called myofibrils. So you saw that on that previous picture. The myofibrils are made of thick filaments and thin filaments. Thin filaments are made of actin, it's a protein. Thick filaments are made of myosin, another protein. So these myofibrils, which are made of thick and thin and thick and thin and thick and thin filaments, are what make that striated appearance for our muscle fibers. That's what gives our muscle fibers those stripes, so to speak. So here you see what I'm talking about. Here is a muscle fiber with all of its nuclei. Inside of that fiber, see all those dots? Those dots are the myofibrils. If we pull out a myofibril and we look at it, we see that it's made of thick, or these are thin and thick and thin and thick filaments all the way down. And that is, again, what's making our cell appear striped. The sarcolemma is that outer membrane. The sarcoplasm is that inner yellow stuff that you see. And the sarcoplasmic reticulum is this blue kind of spiderweb network. Has a very specialized structure that we're going to get into. So you can pause the video and go back and answer these questions referring to what we just described. So let's talk about sarcomeres. A sarcomere is a functional unit within that muscle fiber. This is the actual contractile unit. It's made of those thick and thin filaments. So sarcomeres make up myofibrils. Wow, a lot of vocabulary. Go back and re-listen to it if you need to. Sarcomeres make up myofibrils. Myofibrils are the filaments inside a muscle fiber. So let's talk about all of the parts of a sarcomere. You have something called a Z line, an M line, an H band, an A band, an I band. These terms mean nothing to you without actually seeing what they are. So let's go to the next slide where you can see what they are. This is my myofibril. if you remember, the myofibril is made of thin filaments and thick filaments. The thin filaments are made of actin. The thick filaments are made of myosin. The sarcomere itself is the entire structure from Z-line to Z-line. So Z-line to Z-line. The sarcomere extends from one Z-line to another Z-line. The Z-line is the root of the thin filaments. So the actin filaments are anchored into that Z-line. The thick filaments are anchored to an M-line or midline. This M-line is the middle of our sarcomere. If you notice, our thick and thin filaments overlap. It's called the zone of overlap. You will understand this a little better once we look at how a muscle cell contracts. The A band is the entire area of darker thick filaments, and the I band is our entire area of thin filaments. So let's look at the overall structure of these thin filaments. As I mentioned, they're made of actin and they are attached to that Z-line. So there's actually three main proteins. Actin's the major one. Actin is kind of twisted together. There's also something called tropomyosin and troponin. Tropomyosin covers what we call an active site. The active site is the binding site for myosin on the actin molecule. Again, you'll see this. This will make more sense when you see the picture. Troponin binds to the tropomyosin, and it basically holds the tropomyosin in place. And this is where calcium binds. So let's look at this whole picture. Here you see this twisted actin filament. Again, this is the thin filament. This is attached to the Z-line of our myofibril, of our sarcomere. You have these tropomyosin molecules, the tropomyosin protein. Again, that's where the myosin's going to bind. And the troponin are these little kind of knobs that are blocking that tropomyosin long filament. So the troponin is there. That's where the calcium's going to bind. That calcium is then going to move that troponin and reveal that active site for the myosin to bind. Let's look at the myosin structure, and then we'll put the whole thing together. So the myosin thick filament has a tail and a head. The tails point towards the M-line, towards that middle area of the sarcomere. The myosin head has protein subunits that bind to the thin filament. It binds to the thin filament on those tropomyosin active sites that I just mentioned. So the thick filament is what's going to attach to the thin filament. Here you see the whole structure of this thick filament. They have a head, or they have a tail and a head. And that head, I don't know if you can see my arm, the head kind of flexes like this. It moves. And that little movement is what makes the entire muscle cell shorter, and that's what contracts your muscle cell. That's what's going to move your muscles, pull your bones, and move your bones. So, I don't believe it doesn't have it in here. I'm not gonna flip forward that much. Sorry. I want you to see how they fit together, but that picture is not included. We'll get there, I guess. So let's talk about other parts of the muscle cell. So we've talked about the thick filaments and the thin filaments. Now let's talk about the sarcolemma and the T-tubules. So an uneven, just as a little bit of review from chapter 3, it was covered briefly, an uneven charge distribution on the sarcolemma is called a membrane potential. This is a hugely important concept. If there is a change in the membrane potential, which is a good thing, there's this charge distribution that travels along the T-tubules. So you might be saying, well, what's a T-tubule? I'm going to show you, all right? So this is continuous with the sarcolemma, and it forms a passageway into the cell. It basically surrounds the SR, the sarcoplasmic reticulum. So the sarcoplasmic reticulum is that, remember, it was that blue kind of spider web network inside the cell. It surrounds each myofibril and it touches these T-tubules. And there's calcium in the sarcolemma. So this is where the beginning of muscle contraction occurs. If there's a change in membrane potential, that membrane potential travels into these T-tubules. These T-tubules are like little valleys or canyons on the outside of the cell membrane of the sarcolemma. So that membrane potential can now come into the cell, touch the sarcoplasmic reticulum, and the sarcoplasmic reticulum now can release calcium. Here's the whole process. Here's our sarcolemma, this outer plasma membrane. Here you see the T-tubules coming in. They're just invaginations, or like little canyons or valleys coming in from that outer cell membrane, touching our blue, in this picture, sarcoplasmic reticulum. And so that membrane potential, or that charge, can travel from like a nerve cell that's telling you to move, travel down the sarcolemma into the T-tubule, touch the SR. SR can release calcium. Once the calcium's released, now the contraction has begun. So you can pause the video, go back, and answer these 3 questions. Let's talk about how muscle fibers contract. It all comes from what we call an action potential from a motor neuron. There's a motor neuron that's in charge of moving every single muscle of your body. Every single muscle cell of your body is associated with a motor neuron. Where they come together is called an NMJ, or a neuromuscular junction. The neuromuscular junction is where the end of a nerve cell and what we call a motor end plate of a muscle fiber interact. Other ingredients of this neuromuscular junction include a neurotransmitter. There has to be some type of cellular or chemical signal that's being sent from the nerve cell to the muscle cell. Usually in muscles, skeletal muscles, it's acetylcholine, or ACH. ACH is basically waiting at the end of the nerve cell to be released. So this action potential travels down the nerve cell, releases acetylcholine at the end of the nerve cell. That nerve cell receives, or the muscle cell receives the acetylcholine, has little receptors on it, and then that membrane potential can travel into the sarcolemma and go down the T-tubules and affect the sarcoplasmic reticulum to release calcium, so on. So ACH, acetylcholine, as I mentioned, comes out of what's called the synaptic cleft of our motor neuron, and it binds to special receptors on our muscle cell, as I was just saying. That membrane potential now changes. Once the acetylcholine is bound to the muscle fiber, this causes what's called a polarization effect. And the sodium channels on our muscle cell now open. And when the sodium rushes in, our membrane potential changes, and that is what's going to travel down the sarcolemma to the T-tubules, telling the sarcoplasmic reticulum to release calcium. This rush of positive sodium ions goes into the sarcoplasm, generating this action potential, changing the membrane potential of our muscle cell. Acetylcholinesterase, ACHE, waits in that synapse, in that area where the neuron and the muscle cell are near each other. ACHE, acetylcholinesterase, is waiting there to gobble up and recycle that acetylcholine, so it's not just sitting there constantly stimulating the muscle cell. So as that muscle cell moves down the sarcolemma, it gets to the T-tubules. The T-tubules are telling the SR, the sarcoplasmic reticulum, go ahead and start to release calcium, and that release of calcium triggers the muscle contraction. Here's the whole thing. So it might make more sense when you see it visually. Some of you are visual learners, and I strongly encourage you to go out into YouTube and look for videos of action potentials type in neuromuscular junction, and you will find a ton showing you what exactly is happening here. So here's our motor neuron. It's hovering just above our muscle fiber. It's not actually, it's not actually touching the muscle fiber. It's hovering there. And what you see is this motor end plate. This is part of the axon, part of the end of the nerve cell right here, this blue. So on all of these pictures, the blue is from the neuron and the yellow is representing the muscle cell. Notice that they don't actually touch. There's a gap. It's called a synapse. So these vesicles contain acetylcholine, and as the action potential travels down our nerve cell, you can see that our nerve cell's turning red. That's telling us that there's an action potential there. That action potential triggers the release of acetylcholine. The acetylcholine now binds. You can see it here in a couple instances, that those little red bubbles binding to those little gray pieces. And as acetylcholine binds, it's causing sodium to rush in. As sodium rushes in, that's going to trigger the change in membrane potential on the actual cell membrane or sarcolemma of our muscle cell. That's going to travel into the T-tubules. The T-tubules tell the sarcoplasmic reticulum, release calcium. Acetylcholinesterase is sitting right here in that synapse, waiting to gobble up and eat the rest of the acetylcholine so that the muscle is not constantly being contracted. So you can pause the video, go back, and answer these questions. Let's talk about this theory called the sliding filament theory. Now, we know in science that a theory is based in scientific evidence. It's based in data. We're calling it a theory because nobody has observed every single muscle contraction to make sure this is how every single muscle contraction occurs. We've seen it enough times to know that this is probably the way all of them occur, but we can't call it fact because we haven't actually seen all of the muscles ever that have ever contracted. So that's why it's called a theory. In this sliding filament theory, remember the structure of our sarcomere. We have this zone of overlap where our thin filaments and thick filaments are hovering over each other. Those myosin heads can dip down and pull those thin filaments. That's the sliding filament theory. It's this idea that the thick filaments or the thin filaments slide past the thick filaments. The H bands and I bands get smaller. The zones of overlap get larger. Those Z lines move closer to each other. In other words, the sarcomere shortens. So here's where it all happens. Here you see Z line to Z line. This is our sarcomere. You have that M line in the middle. This is where all of our thick filaments are rooted. The Z lines where all of our thin filaments are rooted. Notice the zone of overlap here. And when that cell contracts, when those myosin heads contract and they pull those thin filaments in one direction, the sarcomere now gets shorter. The thin filaments move toward one another. Our A band does not change, but our I band does change because that area gets much, much smaller. Our zone of overlap increases. So overall sarcomere length decreases. The myofibrils are shortening. As every myofibril is shortening, that's causing the whole muscle fiber to shorten. The filaments themselves don't shorten. They just start to overlap each other more. They slide past each other, hence sliding filament theory. Here you see a myofibril at rest, and here you see a contracted myofibril. These I bands are just smaller on the contracted myofibril. Again, they're at rest. If we kind of fix it at one end, just like you would on a muscle, right? They're fixed on the ends, they're attached to tendons. You get a much, much shorter muscle myofibril. And imagine if you have all of these myofibrils stacked on top of one another, well, you're going to get a dramatic change in length. So this tension is transmitted to bone. The myofilaments are attached at the end of a muscle fiber, and the muscle fibers are attached to tendons on bones. So as that sarcomere contracts, the result is a shortened muscle fiber, and that's pulling on the tendon, that's pulling on the bone, that's causing the movement. So here you see the whole process. I love this picture. You can see the myofibrils inside a muscle fiber, right? All of these are individual sarcomeres stacked on top of each other. And all of the endomysium fuses at the ends to form this tendon, right? And you can imagine as these guys are all shortening, that the entire muscle fiber would pull on this tendon, making that muscle move the bone. So again, contraction is an active process. ATP is the power source of this. You use ATP energy for everything. The strength of the contraction depends on how many muscle fibers are activated. So in other words, when you flex your bicep, not every single muscle fiber in your biceps is actually contracting at one time. They kind of alternate. The more powerful the contraction, well, that just means more muscle fibers are being contracted at that time. Muscle lengthening is not an active process. It's just returning back to length because of gravity, maybe because there's a contraction by an opposing muscle group like biceps, triceps, right? And there's also some elasticity in the tissues that's going to recoil or bounce your muscles back. So here you see the antagonistic approach where your bicep muscle relaxes and your tricep muscle contracts, and then the opposing where your tricep muscle relaxes and your bicep muscle contracts for flexion and extension. So you can pause the video here and go back and answer those 3 questions. Let's talk about myosin at rest. At rest, myosin is energized. Myosin has potential energy when it is resting. That ATP is bound to the myosin and it's kind of sitting in this locked and ready position. And ATP, ADP, and phosphate are bound to that head. So they're ready for ATP to break down. So let's talk about this contraction cycle. Remember, we left off with calcium being released from the sarcoplasmic reticulum. So now we have to look at what's actually happening on the myofibril that's causing this contraction. So that neuromuscular junction ultimately depolarizes the muscle fiber, causing sodium to rush in. That's causing the calcium to be released from the sarcoplasmic reticulum. So once calcium arrives in that zone of overlap, the active sites can be exposed. The calcium ions are going to bind to troponin. Troponin will now move. Once the troponin moves, it's revealing what we call the active site on the myosin head. So now the myosin can attach to our actin molecule. Remember, our actin is sitting there, and it's the active site's being blocked by the troponin. Once the calcium binds, troponin moves, and now the myosin can bind to the tropomyosin. So it's what we call cross-bridge forms. That's the actual meeting of that myosin head and the thin filament. The myosin head will pivot. ADP and phosphate is released with that little pivot, that little movement, that shortening. And our kinetic energy is released, so our ATP is used. When the cross bridge is detached, ATP binds, causing that myosin to be released. So a new ATP is needed for the myosin heads to detach. Myosin will be reactivated when it splits that ADP into ATP. So again, it uses energy to recharge that myosin. Detaching is passive, but to recharge that myosin, it has to be activated again. That whole cycle repeats as long as ATP and calcium are available. So the end of the contraction cycle is when a stimulus from the motor neuron stops. Acetylcholine breaks down because of acetylcholinesterase. The action potential stops in the sarcoplasmic in the sarcolemma and the T-tubules. Sarcoplasmic reticulum gets all the calcium out of that zone of overlap area. The troponin molecules go back to blocking the tropomyosin, and the tropomyosin's covering the active site on the actin, and cross bridges can no longer form. So here you see the entire contraction cycle. Here's a resting sarcomere. And in this resting sarcomere, again, you have ADP and P just waiting in that myosin head. Calcium's being released. So what calcium does is it binds to the troponin. You see it here, binding to the troponin. And once it binds to the troponin can move, and now that cross bridge can form. Now our myosin head can touch our active site on the actin. That's going to cause that ADP to be released and that myosin head to pivot. Now a new ATP is needed for that myosin head to detach. And then for the myosin to be reactivated, that ATP is converted into ADP plus P for potential energy, and our entire process can start again. So you can pause the video, go back, listen, and answer those questions. So let's review all of these events. This is a pretty thick physiology, thick chapter. There's lots of things happening here. Good review. Skeletal muscles are stimulated by a motor neuron. The action potential causes the release of acetylcholine. The acetylcholine binds to receptors, opening the sodium channels. That's going to cause an action potential down the sarcolemma and down the T-tubules. The T-tubules will then cause the release of calcium from the SR. The calcium will now allow for the thick and thin filaments to interact. And now the entire skeletal muscle can shorten, producing tension on the tendons, moving the bones, etc. Here's the whole thing in picture form. Have our motor neuron touching all of the myofibrils here. Here's our acetylcholine being released. This whole process is called excitation. The process is called excitation-contraction coupling. In other words, as soon as that acetylcholine is released, there's no way to stop the muscle from contracting. It's going to contract. They go together. Excitation and contraction will always occur together. So this excitation travels down the T-tubules, touching the SR, causing the calcium to be released. Calcium is going to trigger this contraction cycle. Our muscle fiber can contract, the tension's produced, the muscle fiber shortens, we get movement. Let's talk about some variances here. One's called a muscle twitch. Muscle twitch is a single stimulus contraction relaxation cycle. You've experienced twitches all the time, right? Sometimes you get that eye twitch or maybe a calf twitch. It will depend on the muscle type and location and external, internal conditions. But if you look at a normal twitch, like a twitch of an eye muscle, it's very, very quick and it's producing a lot of tension very quickly. If you get a twitch in your calf muscle, sometimes this happens, it can last for a much longer period of time. This is in milliseconds. So it can last up to 1 second. Or it can generate, or I should say, and it generates the same amount of tension as an eye twitch. It just does it for a longer period of time. So it might feel a little bit more uncomfortable. There are phases to a muscle twitch. There's a latent period, there's a contraction phase, and a relaxation phase. The latent period is where no tension develops. This is kind of the calm before the storm. This is when the tension is going to start kind of getting ready to happen. This is as that action potential is moving down that sarcolemma and the calcium is being released. So it's not quite tension producing yet. The contraction phase is when that calcium binds to the troponin, that cross-bridge contraction, excitation-contraction coupling happens. And then the relaxation phase is at the end of the twitch as the calcium levels fall and the cross-bridges detach. So here you see the, amount of time needed for twitch. This is, again, of the calf muscle. It's just easier to look at something that produces more time. And the reason the calf muscle takes more time for a twitch is because it's a bigger muscle. An eye muscle is much smaller than a calf muscle. There's also something called wave summation. Wave summation is going to occur with a second stimulus before the end of the relaxation period. So in other words, it's going to increase tension. So it contracts and boom, you're hit with another excitation, and boom, you're hit with another one and another one. And so it's this buildup of tension over time because the muscle just keeps being bombarded with action potentials. And one very, very powerful contraction can be produced. It's called wave summation. So twitch after twitch after twitch after twitch after twitch, it's going to build the tension. There's also something called incomplete tetanus. Incomplete tetanus is a cycle of contraction and relaxation producing almost peak tension. Almost peak tension. So again, contract, relax, contract, relax, contract. It's kind of like wave summation, but you get to a peak that's not at maximum tension. Incomplete tetanus, this is where you have a very high stimulus frequency Calcium is never taken back into the SR, so you have this continuous contraction producing peak tension. It's called a complete tetanus. So contract, 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 contract. This is more muscle cells, more muscle fibers being recruited until, oh my gosh, you have complete tetanus. A real-world example of complete tetanus would be like a Charlie horse, right? This is where all your muscle cells in that particular area, maybe your calf or your foot or wherever, are cramping so much that you reach incredible pain. So you can pause the video, go back and answer these 3 questions. Moving on, muscle fibers and motor units. The skeletal muscle is made of several thousand muscle fibers. This hopefully was made clear when we looked at those pictures earlier in the chapter. A motor unit is an area that's controlled by one single motor neuron. It's all the muscle fibers that are controlled by one motor neuron. And the size of this motor unit indicates the precision of movement. Smaller motor units for more precise movements. So something like your hands that need to move very succinctly are controlled by several motor units. Something like your face, which makes lots of weird shapes by moving the muscles, controlled by several motor units. Your back? No. Your back does not have that many motor units on it because your back doesn't really move in precision. So motor fibers of different motor units can be intermingled. There's something called recruitment. In recruitment, what you're doing is, or what your body's doing, is just picking a few muscle fibers to activate it first, and then more, and then more, and then more, increasing tension. So maybe at first you go to pick something up and you're not sure how heavy it is, but you go to pick it up and you can't move it at first, but you keep trying and you keep trying and you keep trying. What that is doing is your motor neurons are saying, we need more biceps, we need more biceps, we need more biceps. So it's basically, sorry about that, basically recruiting more motor units, getting more of these muscle fibers to be activated so that you can finally pick up that box. So skeletal muscles are composed of all of those. Whoops, we just did that. Let's go here. How about we talk about muscle tone? We know what tone is, right? I want to go to the gym. I don't really want to get big. I just want to tone up. Well, what you're saying is you want a resting tone to your muscles. In other words, your muscles are going to remain slightly contracted. That's what tone is all about. Some motor units are active even when the muscle is not contracting. This is going to tense and firm your muscles. It's called muscle tone. It's a real terminology. There's subconscious process where your motor units are being recruited and being active even when you think that you're resting. This is why some people look like they're flexing even when they're not. And you can train your body to do this. You just practice it. You practice holding your abs in, or you practice flexing your biceps. And over time, that tone will increase and your resting tone will be there even if you're not consciously thinking about it. You can pause the video, go back and answer these 3 questions. Let's talk about different types of contractions. All contractions are not created equal. You have an isotonic contraction. This is where the tension rises and the skeletal muscle length can change. Tension rises until it exceeds the load, and then as the muscle shortens, the tension remains constant. So in other words, an isotonic contraction is when you're picking something up and you're actually moving your muscles. And here you see this 2-kilogram weight is being lifted by that muscle. An isometric contraction is where the muscle length stays the same. Your muscle fibers shorten, but your connective tissue stretches. So tension does not exceed the load. This would be like if I were standing here and I'm flexing I'm sitting here actually, but I'm sitting here and I'm flexing my biceps even though I'm holding them down at my side. But you can do that. You can flex your muscles without actually moving. That's an isometric contraction. Here you go. The muscle is contracted, but instead it's the connective tissue on the end that's stretching, it's not producing movement of that 6-kilogram weight. You can pause the video, go back and answer those 3 questions. Let's talk about how ATP comes about in muscle fibers. At rest, the mitochondria will produce lots and lots of ATP. The ATP comes mostly from fatty acid metabolism and glucose. Don't worry, we'll talk about metabolism later. ATP can also be used to create creatine phosphate. Creatine phosphate comes from creatine. It's just a protein. And we can also create ATP from glycogen. Glycogen is the stored form of glucose. With moderate activity, you're going to increase the demand for ATP. Say I'm in my basement right now. Say I need to go upstairs and go get more coffee, right? If I do that, that's going to increase my demand for ATP more than I'm just sitting here. So ATP is produced through aerobic respiration with oxygen. It creates pyruvate in the mitochondria, and that 6-carbon glucose can then be broken down into the pyruvate and so on and so forth. We'll get into all the details eventually. There's no fatigue until your glycogen, lipid, and amino acid reserves are exhausted. So, in other words, your body can keep going even if you feel like you can't go anymore. It's a mind game, which I understand completely as I'm training for a marathon and I go out for a 20-mile run and I think, oh my God, I can't run 20 miles. But I can. You do it. You just push your body. So that's a side note. Let's take a look here at what's going on. Your blood vessels are carrying glucose, oxygen, fatty acids. These are all coming from your respiratory and digestive tracts, right? Glucose comes into our muscle cells, can be converted into glycogen and be stored later, or it can be used to make ATP or creatine phosphate. These molecules can go into the mitochondria and produce lots of ATP as well as carbon dioxide, which we exhale. As we rise our activity levels, our oxygen demand increases. Our body might start to use fatty acids more instead of glucose. Notice our body's not bringing glucose in from the blood while we're doing moderate activity. It may be doing fatty acid metabolism, but any glucose that's being used is the glucose that's already in the cell. So our glycogen is being broken down into glucose, and that glucose can then be converted into pyruvate, go into cellular respiration, release carbon dioxide, and create lots of ATP. This ATP goes directly to our thick filaments, which need ATP in order to contract and reload, so to speak. So at peak ATP production, this is when our mitochondria are producing ATP at a maximum rate. Only 1/3 of the ATP produced by aerobic means. The rest of it is anaerobic. The remaining 2/3 are produced without oxygen. This is just converting glucose to pyruvate and pyruvate to lactic acid. The lactic acid will dissociate and create a hydrogen ion, which lowers the pH. That's why it's an acid. Lowering the pH is going to affect your enzymes, preventing your muscle cells from contracting and resulting in fatigue. Guess what? You can build up tolerance to this, and you can also train your body to not create as much lactic acid by training. This is why I can go out and run 20 miles, but you may not be able to do that because you're going to be feeling the burn a little bit too much from all of that lactic acid. Don't get me wrong. I'm sore after a 20-mile run, but I'm not debilitated. So this is the process here. Glycogen being converted into glucose, being converted to pyruvate, being converted into lactic acid. That lactic acid is dissociating. This hydrogen ion's lowering the pH, tiring your muscles out. This is where you say, but I can't do it. I feel the burn too much. You can keep going. You physically have the energy there. You just have to train your body to use the oxygen more efficiently. So there is something called the Cori cycle. This is where the lactate that's going back into the blood is recycled by the liver. The amount of oxygen that's required to return your body back to normal oxygen levels is called an oxygen debt. As you are working at peak performance, your body's going to use more oxygen so that when you finish working out, you're going to have a little bit of oxygen debt. This is why your heart rate and respiration rate stay elevated even after you finish exercising, because you need to continue to bring in more oxygen to replace that oxygen debt. Here is the Cori cycle, all of that lactate being released from the lactic acid, going to the liver, where it's converted back into pyruvate, back into glucose, which goes back into the liver cell. It can also be converted into glycogen in the liver as well. You can pause the video, go back and review if you need to. Let's talk about hypertrophy, muscle hypertrophy. This is when your muscles get bigger, causing your muscles to develop more mitochondria, more glycolytic enzymes, more glycogen reserves, more myofibrils, enlarging the whole muscle. increasing your muscle strength. This is when you work out and you get big muscles. It's called muscle hypertrophy, like this guy. I think I know him. Muscle atrophy is the opposite. This is where you have a lack of stimulation. This is where you're not working out. It's going to cause you to lose muscle mass, lose muscle tone, make your muscle fibers get smaller. You're also going to lose muscle strength. Atrophy also happens with natural aging. It can be temporary if you were immobilized with a cast or something like that. Dying muscle fibers are not replaced, so atrophy can actually be permanent. If you suffer an injury that requires your muscle or that particular area of your body to be immobilized for an extended period of time, you might not be able to replace the muscles in that area, so you'll have to adapt different ways of moving. Like this guy, he was probably, I don't know, I'm just guessing, maybe in a car accident, and his leg muscles never fully recovered, so he has to be in a wheelchair. Clinical conditions like polio, that's where there's a virus that actually attacks the motor neurons, and that's going to cause muscle atrophy because the motor neurons aren't stimulating the muscles. Or tetanus. Tetanus is an infection caused by bacteria that live in low oxygen environments, and they release a toxin, which again is inhibiting your motor neurons. That's going to result in, it stimulates your motor neurons and prevents them from being inhibited. So this is why you get those powerful contractions. With tetanus, can be very deadly depending on where in the body it is introduced. Botulism is another one. This is going to prevent acetylcholine release of the neurotransmitter. So if that muscle is releasing acetylcholine to trigger the polarization of your muscle cell, well, if it doesn't release acetylcholine, then the muscle cell won't release calcium, it won't contract, right? So your muscles will be paralyzed. There's also myasthenia gravis. This is a disease that's characterized by a loss of ACH receptors on your muscle cell. So again, your muscles are just going to get progressively weaker, not responding to the ACH because they don't have ACH receptors. This is just a look at the overall diseases and what they do. All right, keep on moving. Muscle cells that do not have ATP. ATP is required for our muscle cells to contract. Lack of nutrients, lack of oxygen, lack of ATP can result in, well, no ATP. It can result in calcium not being pumped back into the SR. It can result in those cross bridges not detaching. It can result in a sustained contraction where because those cross bridges don't detach, they just stay attached and stay tight. This occurs after death. Once the ATP is gone, you get to a phase called rigor mortis, and this is because your sarcoplasmic reticulum is no longer releasing calcium, or all the calcium is released because the sarcoplasmic reticulum is deteriorated, and then there's no more ATP available, so those myosin heads are contracted and stay tight, which is why bodies get stiff after a few hours. You can pause the video, go back and answer these 3 questions. Let's talk about the functional organization of our skeletal muscles here. They're about half of our body weight should be muscle. It's more than any other organ system if you're of a healthy weight. It's organized into 2 main classifications: axial muscles, which are supporting the axial skeleton, our head, neck, thorax, abdomen, and then the appendicular muscles, which are our limbs, our arms and legs. There you go. Normal body weight should be mostly bones, skin, hair, nails, and muscles. Everything else makes up a much smaller percentage. Here you see the axial muscles on our trunk and the appendicular muscles on the limbs. Muscles have 3 main terms associated with them. Skeletal muscles have these 3 main terms. The origin, which is where it's fixed. The insertion is on the movable end, and then the action is what it does. It's flexing, extending, etc. So here you see our biceps brachii muscle. The origins are usually proximal to the body, so they're usually close to the body. The origin of the biceps is up here on the head of our humerus and the acromial process. The insertion is on the movable part down here. It's on our radius, and the action is flexing. So there's also functional descriptions in terms of their movement. They can be an agonist or a prime mover. This is the one that's primarily responsible for a particular movement. So our biceps is the prime mover in flexing the arm, or in this case, it says bending the elbow. A synergist helps the prime mover, and this would be like the brachioradialis. You can't see the brachioradialis in this picture. The brachioradialis sits right here underneath the biceps brachii, and that's another flexor. It's just kind of helping the biceps brachii. And there's also an antagonist. This is the opposing action. So this would be like the triceps if we're talking about the biceps. Here you see all of them together. There's our brachioradialis muscle, and here's our biceps brachii. Here's our triceps muscle. So this is the prime mover. This is just a synergist helping that action occur, and then the antagonist is going to extend at the elbow instead of flex. Our skeletal muscles are named by the region of the body, by their position, by their origins or number of origins, by their shape. They can also be named by their size or by their action. Wowza. Here are the terms. Do you have to have this memorized? No. I would rather you spend your time spent learning the actual muscles than memorizing this list. This is just showing you some examples. So you can pause the video, go back and answer these 3 questions. Let's talk about flexors and extensors. The videos, by the way, these can all be accessed by going into the Mastering A&P website that came with your textbook. I've posted the code to the course in an announcement, or it may be in the syllabus. I think I posted it as an announcement. All right. So flexion is the movement that's going to reduce the angle between structures. So again, you're bringing something toward your body. You're flexing your arm. Your legs are going to flex backwards as you kind of kick your butt. Extension is going to be the opposite, and hyperextension is going beyond anatomical position. So hyperextending your wrist would be something like this. Hyperextending your arms is doing like Superman and pushing your arms behind your body. Here they all are. Nice movements for you. You can see flexing the head, extending the head. All of us are probably extending our heads right now. Maybe you're flexing it a little bit if you're drooping. Hyperextension is leaning your head back. Same thing. Arms and legs move in opposite directions from each other. There's also lateral flexion, where you can kind of bend from side to side, or dorsiflexion when you refer to your feet. Plantar flexion, toes pointing downward. Dorsiflexion is pointing your toes like up towards your knees. Lateral flexion, as you see here, dorsiplantar flexion. Abduction and adduction are just moving your arms away from your body, and abduction and adduction is adding it back to your body. Here you see them. You can do it with your fingers. Abduction, adduction. Circumduction is going around. Really, the only joint that can do this is your shoulder can circumduct. I can't do it with my left shoulder because I broke it a while ago. So I'm not very good at circumducting. There you see your circumducting. And you can pause the video, go back, answer these questions. Couple more body movement terms here. You got rotation. You can rotate your trunk, rotate your limbs, right? Move them around. You can do left rotation, right rotation. You can also do medial rotation, where you move your arms inward. I don't know if you can see. Moving my arm inward like this. Or lateral rotation, moving outward. Opposing movement. Pronation and supination. Supination, pronation. And yep, pronation and supination is specific to the forearm. Opposition specific to the thumb. Inversion and eversion deal with your feet. I'm doing it with my foot, but obviously you can't see it right now. Inversion's moving your foot inward. Eversion's pushing your foot outward. And then protraction and retraction. Moving your head like this, or your jaw. Or depression, elevation. Right, here's opposition, protraction, retraction, inversion, eversion, depression, elevation. You can pause the video, go back and answer these questions. So let's talk about specific muscles. Yay, I told you this was going to be a long, heavy chapter. Our axial muscles are the majority of our skeletal muscles. 60% of our skeletal muscles are just on the core of our body. They all originate on the axial skeleton. They help position our head, our spinal column, rib cage, assist in breathing, etc. Appendicular muscles, these are all of our arms and leg muscles. There they are. Do you have to know all of them? Yeah, only a few of them are tricky. Most of them you're probably a little bit familiar with if you've maybe been a part of an exercise routine. If you've never exercised before, you have maybe a little bit more learning to do than someone else. Am I going to give you a diagram on a test or quiz? No, I will not give you diagrams. Maybe your lab instructor will. You have to ask her or him. For my part of the class, I may ask you, which of the following is not part of the arm? Which of the following is a flexor of the forearm? Which of the following would you find on the back, etc.? So I'm going to ask you application type questions of these muscles. I'm not going to ask you to point them out on the body. There's your posterior muscles. Pause the video, go back and answer these questions. Let's talk about muscles of the head. And I'm going to go through this super quick again because this is all covered in lab. And also because, like I said, I'm really just asking you big picture type questions on the muscles on your tests and quizzes. So muscles of the head and neck are used in facial expression and for eating. Your muscles on your face are different from the muscles on the rest of your body because they typically originate or insert on each other. They don't originate or insert on bones specifically. So like your orbicularis oculi, orbicularis oris, they're just originating and inserting on the surrounding muscle tissues. And that's why we can get such precise movements of our face, so on and so forth. Do you have to know origin, insertion, and action? No, not for my part of the class. You may have to know it for lab class, though. You just have to know what they are. So which of the following is on the face? Which of the following is used in facial expression? Something like that. Here you see muscles of the head and neck. And you can pause the video, go back and answer these questions. Then you have muscles of the vertebral column. They come in several layers. They originate on the ribs as well as on the vertebrae. They help extend from the sacrum all the way up to the skull. So they cover that whole back area. And they can be very, very long or very, very short, depending on what they are. Again, for my purposes, you just have to know that they are there. You do not have to know specifically their origin, insertion, or action. These questions, you can pause the video, go back, and answer the questions. Coffee break, sorry. Oblique and rectus muscles. Oblique muscles are always at an angle to the body, and rectus muscles are parallel to the body, which makes sense if you think about our abs. You have our rectus abdominis, which are parallel to the body, and then we also have internal and external obliques, which go at an angle to our body. These are the muscles of our thoracic and abdominal region. Again, you just have to know that they are there. You don't have to know specific actions, origins, or insertions. You can pause the video, go back and answer these questions. Muscles originating on the trunk will help move the limbs. They are usually very large and powerful, and they can help hold our appendices, hold our limbs in place. So these would be things like these large pectoral muscles or deltoids, etc., trapezius, all originating on the axial skeleton but inserting on the appendicular skeleton. And this is some questions that you can go back, pause the video, and review. Upper limb muscles, the ones that are closer to the body, again, are stronger and less precise. They can help extend the elbow. The flexion muscles are usually on the anterior or front surface. The extensions are on the posterior surface. And they all have tendons to hold them in place. And here you see our extensors, and here you see our flexors. Flexors on the front. Except on the legs, it's the back, it's opposite. So here you can pause the video, go back and review. The flexors of the knee all originate on the pelvic girdle. They hold our thigh together. These would be our hamstring muscles. You should know the 3 hamstring muscles. That's a good test question there. These are the flexors. On our legs, the flexors are on the back. Extensors are on the front. Extensors are quadriceps muscles. Again, you should know the 4 quadriceps muscles. There they are. All of our extensors are on the front of the leg. So you can pause the video, go back, and answer these questions. Almost done with chapter 6. Our lower leg muscles, the extensors, again, are on the back. So it switches, and there you see them. More extensors from a lateral view. You can pause the video, go back and answer the questions. And oh, that's done. We are done skis for chapter 6. You can go back and review by re-listening to the lecture, completing your reading guide. There's also activities online. As I mentioned, I strongly encourage you to go into internet world and type in animation of action potential, animation of skeletal muscle contraction, and you will find a lot of videos there to help you with your understanding. I like to watch videos when I learn things because it helps. I'm a visual learner, so that really helps me understand what they are. I will try to post any cool, interesting videos that I find in your announcements for the week. Have a good one. Bye.